Go ahead.
We have calibrations for starchal protein phosphorus.
There's
All right, welcome everybody. Good afternoon. Thank you for joining us today for day 2 of the Texas A&M Plant Breeding Symposium. Today we have some very good presentations coming up, but first we'd like to thank DuPont Pioneer for their sponsorship of our symposium once again. They helped provide scholarships for 4 individuals to come from all across the United States to give talks today and to experience the symposium. We will be hearing later from Matthew Mattia from the University of Florida, from Anna Levina from Cornell University, and from Clint Secchi from the University of Georgia. I will warn you guys, unfortunately we may go a little bit over our 1-hour time limit today, so please be aware of that because we're getting started a little bit late. But first we're going to start with Alexandrea Ollhoff. Alex Ollhoff is pursuing her PhD in plant breeding and molecular genetics at the University of Minnesota. Her current research uses genome-wide association and other genomic approaches to dissect complex traits in barley nested association mapping populations. In the future, Alex's career goal is to develop more nutritious crops that also help to reduce the environmental impact of her agriculture through resilience to biotic and abiotic stresses. She earned her BS in biology from the University of Wisconsin-Stevens Point. So, without further ado, let's get started with Alex.
Thank you guys so much for inviting me. I'm excited to have you all here to give me some feedback about research and discuss things. So, this is a picture of Svalbard. It's the world's largest gene bank. And gene banks around the world hold over 7 million accessions of every crop species. They're by far our best genetic resource for sources of disease resistance, drought stress, anything that exists, any kind of genetic diversity that exists for our crops is held within these gene banks. But they're used relatively rarely. Yesterday, one of our speakers mentioned that they had been evaluating over 70,000 accessions of wheat for a number of different traits, and that's probably one of the most comprehensive screening trials that's occurred. One of the other great examples that I know of is in barley, where over 23,000 accessions were screened for Fusarium head blight, and it took 12 years to do. It's very time intensive, very labor intensive. And one of the reasons is because none of it is adapted to a single location. A lot of it looks like this in the field. And you can imagine that it's hard to get any kind of disease rating or even a heading date, a flowering time, on lines that do this. So those are our best resource. We don't usually want to put them into a breeding program that already looks like this. So this is a barley, one of the elite germplasm nurseries. These are lines that are being evaluated for malt quality already. And the Minnesota breeding program is notorious for having a narrow genetic base. Historically, they haven't brought in a lot of diverse germplasm, but there are lots of scenarios where they could be very helpful. Barley, of course, is malted and then used to make beer and other malt products, and it's also used for animal feed. And because malting requires such a specific suite of quality traits, there isn't a ton of diversity that's been introduced. A pedigree of some historic releases shows just how few introductions there have been into the breeding program historically. So the 7 highlighted in yellow are Minnesota releases. It's kind of the poster child for having low diversity. We've all seen this iconic figure from a Tanksley and McCooch paper that shows the loss of diversity through the domestication process, and then again, even more loss through crop improvement. So these are our germplasm collections, and on the far side are our breeding programs. Really, we need a resource that can bridge the two. One solution to this is a nested association mapping population. So we can take germplasm that looks like this and cross it to a single common parent to generate families of lines that we can eventually use to mine traits and alleles that can be incorporated into breeding programs. Nested association mapping, or NAM, populations have been used really extensively in maize to help understand very complex traits. The basic principle is that a number of diverse lines, and in the case of the barley nested association mapping population, we have 91 diverse lines that were crossed to a single common parent. We use the parent Rasmussen because it's high yielding and adapted to the temperate climate of the Midwest. We crossed all 91 lines to a common parent and then derived families from each cross. So there are 91 families of crosses, really 91 biparental populations within the entire population. To give you a snapshot of what the population looks like, this is the number of lines within each family. On some of the lines were actually back-crossed to population, so there are some back-cross 1 derived lines, and there are some F1 derived lines within each family. But a better visual yet is an aerial photo. This is just under half of the population. The whole thing takes up roughly a football field. So the takeaway from this is that with getting a lot of power from diversity, like power of detection, we have a lot of lines, but it's not a practical number of lines. 8,000 lines in the entire population is not always practical for evaluating very complex traits. So we need a way to pare down this population if we want to be able to evaluate it for the traits that it's most powerful to detect. So we go into this with a lot of background knowledge. A really well-studied locus is photoperiod 1. There are 2 main alleles that have been described, I guess. The dominant allele is PPDH1 capital. It's present in barley that is native to the Fertile Crescent, and it allows the barley to flower early enough to escape the heat and drought of summer. The recessive allele is present more commonly in varieties that have been developed in Northern Europe, where there isn't as much pressure to flower very early. So flowering time. I think this sums it up pretty well. It's associated with a lot of important traits. When it says it's associated with most traits, it's referring to plant height, node number, 1,000 kernel weight, number of tillers, and internode length. Those are a lot of really important traits. And in association mapping studies, there are many examples of studies where they're looking for QTL for these traits, but they find PPDH1. And because it's associated with so many traits, it's not necessarily useful for breeding. So this isn't surprising, but how can we use it to design very smart and strategic association mapping or genome-wide association studies that use this prior information? So we can use this as a way to very strategically subsample from the NAM population. So the NAM parents, about just under a third of them carry the recessive PPDH1 allele, including the common parent. So about a third of the families are fixed for this allele. So if we just sample those families that are fixed, what other variation can we detect for other important traits that are often confounded by the variation that we see that's due to PPDH1? So we get to actually look at a lot of data. So the blue families are the ones that carry the recessive late flowering allele, and that allele often also confers higher grain yield. So if we look at the families that carry that allele, though they're fixed for that locus, and it's very important, there's still a lot of variation within those families. So it indicates that there are other genetic loci that are contributing to variation for even heading date. And then if we group the families that carry similar alleles, they actually have significantly different mean heading dates. So even though this allele has been detected and studied in other populations, it says that it's still very important, even within diverse germplasm. And most importantly, the N is 1,543, which is far fewer than 8,000. So if we just consider, so this is the distribution of heading date and the frequency across all the NAM lines. The blue lines are the families that are fixed for the PPDH1 recessive allele. And a lot of the variation for heading date is maintained even within that allele class. And this indicates that there's still variation that could be useful for mapping QTL that contribute to variation for this trait. So my next question would be, how does it contribute, or how does it limit the variation for other traits that we might want to study? And we can get an idea by looking at the same distribution for another trait. So if we consider height, there is some loss of variation on the lower end of height, but really a lot of it is maintained. So this is polarized for the same two alleles, where PPDH1 is blue, and a lot of the range for the height trait is maintained. So even if we have a subsample of the NAM lines, which is, so I said like all 8,000 NAM lines takes up roughly a football field, and that's not really practical for measuring some traits, especially in multiple locations and multiple reps. So if we want to narrow it down, if we use strategies like this, and we can narrow it down to 1,500 lines, that takes up more like this, which is much more practical. So it's a way for us to tap into the diversity of germplasm collections, but have them be adapted to more adapted, not necessarily always adapted. As I've seen in my field, there's still a lot of variation. But it's a way for us to tap into the diversity, have it be at least somewhat adapted to a single environment, and then we can use prior information to subsample from the population, build a really strategic GLOS panel. Yeah. So I would like to thank the many people who helped me come here today, and my training agencies, and yeah, I'm happy to have any questions.
We have time for a couple questions. Raise your hands. Dr. Murphy.
You talked about the narrow window of malting quality traits,
Yes.
and that's part of the reason there's a really narrow genetic base in barley now. How do you feel the increase of microbrews and the kind of widening of that trait quality, beer malt quality trait window will affect the breeding program?
So we've seen it a little bit already. So traditionally the Minnesota breeding program has been a 6-row malting barley program, but we've recently started a 2-row program as well, because craft maltsters and craft brewers are really interested in using 2-row barley. Barley is 2-row or 6-row grain on the spike. And they've also indicated that things that are just outside the normal malt quality window, like they'll try it. They'll try things that are going to turn out, if your beer is going to be a different color or have a different flavor, like they'll give it a try. And so it has, like, RAR. Malting is, RAR is the largest malt house in the world, and it's in Minnesota. And they have just gotten funding to put in, like, this micro malt system, so that we're able to take some of these weird barleys that we have and malt tiny, tiny batches, and actually, like, it's a 1-bottle malt size, and then they have a 1-bottle brew system to go with it. So they can take these weird barleys and malt and brew with them and see how it turns out. So it's kind of expanded a little bit, I guess.
So I assume 2-row and 6-row is probably just a single gene, right? So why do people make such a big deal about 2-row or 6-row?
A lot of it is history. You could probably speak to this too. A lot of it is like history and tradition. And then I don't know enough of like brewing and malting chemistry.
Are they maintained as separate populations, so
They
they show
are.
population structure between the two of them?
Yes, extensively. All mapping studies are done in one or the other and stuff like that. But there's also like, we're trying this thing with CRISPR to change that one gene, to change our best 6-row to a 2-row. So then we'll see how many differences there are other than that gene.
That was a very interesting talk. I really enjoyed it. I'd like to know, what is your ultimate goal with this project? What do you hope to accomplish by the end?
I'm hoping that it makes a good entry point to evaluating diversity that holds like good drought tolerance and things like that. Because right now, so the example that I talked about with fusarium, where there isn't a source of resistance within the breeding program. And having to evaluate 23,000 unadapted lines to find that, there's hardly ever that amount of resource. There are hardly ever the resources to do that. So hopefully having a resource like this would at least be a good place to start when there isn't a good source of resistance.
So this is exploratory, see what you find kind of thing.
But also, like, my thesis is mostly a lot of genome-wide association study.
Okay.
So that will be like how I'm going to show how effective subsampling is.
Sure.
Yeah.
Good.
So Alex gets bonus points for showing Texas Aggie football in your presentation for sure. So Minnesota has a similar program as to what brought you guys here, right? The Travel
Yes,
Awards.
we
Do you want to
do.
explain that to them quickly?
Yes, our plant breeding symposium is April 8th. We have travel scholarships available. They are open on our website. I think it's plantbreedingsymposium.umn.edu. It's on Facebook too. And I hope that some of you will apply and come visit us in Minnesota. Our student presentations are during breakout sessions on the day of our symposium. And we would love to have you.
Is it required you show the University of Minnesota?
Well, I don't know.
The hockey. All right, let's thank Alex one more time for her presentation.